Okay. Am I audible?
Yes, sir.
Yeah. Okay. Very good evening to all my dear friends. Welcome today session. So yesterday we have discussed some of the important area. We have discussed on the topic is chromosome, the structure of the chromosome. Okay. Structure of the chromosome. The screen is full screen is visible, my dear friends.
Yes, sir.
Okay, so this today we are going to continue the yesterday class, that is the number of chromosome number. One second. Okay. So yesterday we have discussed about the number of chromosomes. Chromosome number. Am I audible? The screen is visible.
Yes, sir.
Okay. So number of chromosomes. Yesterday we have discussed about the euploidy. Euploidy means the chromosome set, the sets of the chromosome is completely present in the cell or nucleus. So this presence of whole set of chromosomes we can call as euploidy. Whereas the missing or the one set of the chromosome or the entire set of the chromosomes we can call as what we can say that aneuploidy. Aneuploidy. So euploidy we are discussed yesterday. Right. So euploidy we are discussed yesterday. In this euploid, the chromosomes may be haploid condition or diploid condition or triploid condition, even though tetraploids also. So haploid means one set of chromosomes. The chromosomes contain only one set of chromosomes, we can call as haploids. Generally, we are observed only gametes of the in sexual reproduction. We are observed the female gametes or male gametes which possess one set of chromosomes. So that's why they are haploid chromosomes or haploid. The chromosome state we can call as haploid state. Is it right? And some of the somatic cells are in the vegetative part of the plants, whereas the roots, stem, leaves, or any other vegetative parts contain, there are two sets of the chromosome. The two sets of the chromosome we can call as the chromosome condition, we can call as the diploid condition. So diploid condition means two sets of the chromosomes we are observed in the somatic cells of the animals, whereas the vegetative parts of the plants are vegetative cells of the plants. Then the aneuploidy. The next one we will going to discuss about the aneuploidy. Aneuploidy means whereas the chromosome, the whole set of chromosome is not able to possess, but they loss of one set of chromosome, or even though addition of the one set of or two sets of the chromosome we are observed in the aneuploidy condition. So in aneuploidy condition, so chromosomes we are observed whether extra or missing chromosomes. If two set chromosome, that is diploid condition, but missing of one chromosome we can call as monosomic condition. Monosomic. Mono means one. Monosome. One set is missing. Okay. So 2n minus 1 we can call as monosomic. Then trisomic. Trisomic means we can absorb addition of the one set of chromosome, 2n plus 1. So this we can call as a trisomic condition. Then nullisomic. Nullisomic means 2n minus 2. 2n minus 2. Null means 0. Null means, my dear friends, null means 0 condition. Am I right? So nullisomic, 2n minus 2. Then tetrasomic. Tetra means plus 2. we can observe plus 2 chromosomes. For example, 2n plus n plus n, we can observe that is a 2n plus 2. 2n plus 2, like that. So this we can call as a tetrasomic condition. Tetrasomic conditions. These are all in the part of the aneuploidy condition. Aneuploidy conditions. So this aneuploidy condition leads to mutations of the different cells or different parts of the plants or the living organism. Am I clear? Am I audible?
Yes, sir.
Yes, sir.
Yes, sir.
Yeah. Then after the number of the chromosome, we are going to discuss about the types of the chromosome types. We are observed the chromosomes in the time of where the cell is going to division, whether the mitotic or meiotic division. We are observed these chromosomes, whether the metaphase or anaphase, the chromosomes, the shapes of the chromosomes based on the presence of the centromere in the part of the chromosome. Based on the structure, chromosomes are classified into or divided into 4 types. Chromosomes are classified into or divided into how many types? 4 types. So number 1 metacentric, number 2 submetacentric, number 3 acrocentric, number 4 telocentric, based on the presence of the centromere in the chromosome. So in the position of the centromere, our chromosomes are classified into various types. Number 1 metacentric, number 2 submetacentric, number 3 acrocentric, number 3 telocentric. Is it clear? Based on which position or which parts, my dear friends, chromosomes are classified into?
Position of centromere.
Yeah, centromere, the position of the centromere, where it is situated in the chromosome. In the chromosome, the centromere position, we can classify or are divided into 4 types, whereas the metacentric, in this metacentric chromosome, the position of the centromere in the middle part or center part of the chromosome. And the next one, submetacentric, means the centromere towards the one end or nearer to the one end of the arm of the chromosome or a chromosome part. The 4th one is telocentric. So if the centromere is very near to the end or 1 telomere, we are observed only 1 in this telomere condition, telocentric condition, we are observed only 1 arm only. We are observed only 1 arm. So these are the 4 types. We will show, I will show the next slide. I hope this next slide is visible. Metacentric, submetacentric, chromosome structures. Chromosome structures you are observed in this slide? No. Okay. Now, okay. I hope you clearly visible the structures or the types of the chromosomes, So first one is the metacentric chromosome. This metacentric chromosome, the centromere position, the middle part of the chromosome, so equal length of the, between this centromere part to the end point of the chromosome or end part of the arm, these are the equal length or the middle part of the centromere. This type of chromosome we can call as metacentric chromosome. Whereas the submetacentric chromosome, the centromere is not in the middle part, it moves towards the one end. That we can call as submetacentric chromosome. This is the second type of chromosome. The third one is the acrocentric chromosome. This acrocentric chromosome, the centromere very nearer to the telomere or end of the chromosome or end of the arm. Then the last one is the telocentric. In this telocentric, we are observed only 1 arm. So the centromere is very clearly we are observed one end of the chromosome. So these are the types of the chromosomes. We are observed in the anaphase of the mitotic division in vegetative cells, whereas we are observed in the meiotic cell division in the flower bud by the use of flower buds. In meiosis, the anaphase condition, we are observed these type of chromosomes. So these type of chromosomes we are observed in which phase, my dear friends? Anaphase. Anaphase. Am I audible?
Yes, sir.
Yes, sir.
Okay, very good. Then after the structure of the chromosome, so we will going to move one of the important area, whether all chromosomes are in the same, or the size of the chromosomes are same, or the chromosome number is equal number of the all species. So these are the questions are arise. So based on the length of the chromosome or the size of the chromosomes, we are observed then more than the very highly largest chromosomes we are observed in the different living organisms like amphibians or insects or Diptera, insects and other plants like Acetabularria, different types of the chromosomes we are observed. That largest or long thread-like structures, long chromosomes we can call as GAINT chromosomes. What we can say that? GAINT chromosomes. These GAINT chromosomes are we are observed in the animals. By the plants, three types of the GAINT chromosomes we are observed. The first one is the Polythene chromosomes. The second one is the lamp brush chromosomes, and the third one is B chromosomes. B for Bombay. Am I clear? So how many types of the GAINT chromosomes we are observed in different living organisms?
3 types.
3 types. Polytechnic chromosome, lamp brush chromosome, and the third one is the B chromosome. Third one is the B chromosome. And my dear friends, the Polytechnic chromosomes also we can call as Polytechnic chromosomes also we can call as a salivary gland chromosome. Salivary gland chromosome. Salivary gland chromosomes. And this Polytechnic chromosome first identified by Balbioni. Who identified these Polytechnic chromosomes, my dear friends? Balbioni. This GAINT chromosome, Polytechnic chromosomes are the one type of the GAINT chromosome or GAINT chromosome, which is first identified and explained by Balbioni. Who explained, my dear friends? Balbioni in 1881. Balbioni, 1881, clearly explained about this GAINT chromosome, that is Polytechnic chromosome, which is identified in the salivary glands of some of the insects' salivary glands. Some of the insects' salivary glands. Okay, then some of the friends is asking me, sir, how these GAINT chromosomes are arise or occurred, how these GAINT chromosomes are formed in these salivary glands of the insects or some of the other living organisms. So just because of, my dear friend, these GAINT chromosomes or GAINT chromosomes are formed due to the replication of the DNA many times within a nucleus or within the certain area. So that's why the GAINT chromosomes are formed or GAINT chromosomes are produced. In these Polytechnic chromosomes, I will show the structure of the Polytechnic chromosome. Next slide. Okay. So this structure, I hope this Polytechnic chromosome structure you are observed in this slide. This is the Polytechnic chromosome bands, you know. So number of bands we are observed inside the nuclear region. So these are IB is interbands, and these are the bands, and this is the puff or puff ball, you know, puff. Whenever we are using powder or during the makeup, using puff, you know. So like that puff, a smooth structure, a puff-like structure we are observed in the chromosome. The larger swollen area we can call as puff, you know. So these are the puff. And this ring we can also call as Balbioni ring. This puff is also we can call as Balbioni ring. Why we can say this Balbioni ring? Just because of these chromosomes first identified and explained by The scientist, Balbioni. So that's why this ring we can call as Balbioni ring. Balbioni ring. These polytenic chromosomes contain a large number of bands inside the chromosome, and also the bands, some of the areas are very dark region bands we are observed, and some of the regions we are observed the light regions, or they are the band width, the distance between the bands is some of the it leaves the space between the bands. The bands looks like a very light color regions, and the puff regions or the darker regions we are observed the very coiling DNA highly coiling regions that are the dark band regions that we can call as band regions. And wherever the uncoiled regions, wherever the DNA uncoiled regions are light color regions we are observed between the bands, that area is very useful for the RNA synthesis, or what we can say the translation and translation process. We are observed on that light color regions are interband regions. Okay, the interband regions or uncoiled regions are very important for the genetic recombinations or gene activity. Okay, so these salivary glands and guts of some flies. Flies means insects, even the protozoans and some of the plant somatic cells we are observed these polytenic chromosomes. These polytenic chromosomes also we can call as salivary gland chromosome. Why we can call salivary gland chromosome? First identified by Balbioni in 1881 in insects salivary glands. Insects salivary glands. So this is the structure shown about the polytenic chromosomes. This is the structure shown about the polytenic chromosomes. So this polytenic chromosomes clearly shows, say IB means, I hope you follow my class, my dear friends.
Yes, sir.
Okay, so this IB means interbands, B means bands. So these bands are in band region, the DNA is coiling very folding, so many folds, so many coils we are observed in the band region, whereas interband region, the uncoiled region we are observed. So this is the interband region, and this is the area is the band region. And the puff region also we can observe in the chromosome of the polytenic chromosome. This is the puff. So this ring we can also call, according to Balbioni, this ring we can also call Balbioni ring. So this is about the polytenic chromosomes. And the second type of chromosomes, and my dear friends, before going to second type of chromosome, these polytenic chromosomes are very important. So during the metaphase and during the mitotic division, even though the interface and prophase of the mitotic cell division, the chromosome is in haploid condition. So haploid conditioned chromosomes like that. So the polytenic chromosomes are always haploid conditions like that. So these polytenic chromosomes identified even though Drosophila melanogaster, Drosophila melanogaster. So these Drosophila melanogaster salivary glands show this polytenic chromosomes. Okay, poly means many. Ardhamaitunda? Poly means many. Many means so many. So the bands, number of the DNA, so many replications are occurred in the same nucleus. So that's why the chromosomes is going to in the center part of the chromosome, or in middle part of the chromosome, or one part of the chromosome area is going to swollen to form a puff-like aperture or puff-like structure we can call as Balbioni ring. And my dear friends, remember these chromosomes always in which condition? Haploid condition. And they are maternal, even though maternal and paternal, they are alike. They are very closer chromosomes. They are very closer in the nucleus. These maternal chromosome and paternal chromosomes, they are alike or they are very closer in the nuclear structure. This is about the Polytechnic chromosomes. Then the next one, the second type of chromosome, that is the lamp brush chromosome. What is that? Lamp brush chromosome. Okay, so the second chromosome, lamp brush chromosome, is another second one chromosome which is identified in 1982. These lamp brush chromosomes identified in which year? 1982. 1982 identified or observed in the amphibians of the amphibians like salamander, some other amphibians, oocytes, amphibian oocytes. These lamp brush chromosomes are observed. Why this name has given these chromosomes lamp brush? Why this term has given to these chromosomes? Just because of they are looks like a the brush, like a cleaning brush of the bottles. So bottle cleaning brushes like that. So these chromosomes looks alike or looks like that. So that's why these chromosomes named given that lamp brush chromosomes. Okay, these are the large and have extensive DNA looping. So number of loops are present in these lamp brush chromosomes, and this is to be extended uncoiled version of the normal meiotic chromosomes. So in last chromosome, in previous chromosome, that is the Polytechnic chromosomes, we are observed in the vegetative cells. So in mitotic cell division, they are also in haploid conditions. But whereas these lamp brush chromosomes, we are observed in the oocytes of the amphibians. So that's why this type of cell division occurred in this type of chromosomes. Meiotic cell division. Why? Because these lamp brush chromosomes are occurred in only oocytes. That is nothing but reproductory cells. So these reproductory cells, this type of cell division has occurred. Meiosis or meiotic cell division we are observed. So these normal meiotic chromosomes are lamp brush chromosomes. So these chromosomes are observed in the vertebrate oocytes. Oocytes means germ cells. So nothing but reproductory cells, you know. So the germ cells, meiotic cell division we are observed. So they are found in amphibians oocytes like salamander. The best examples of the amphibian is salamander, you know. Have you ever heard the term salamander, my dear friends?
Yes, sir.
So salamander. So anybody can ask me, sir, oocytes means this is female organism, female salamander, you know, female oocytes. Amphibians, the best example of amphibian, salamander. In salamander, female oocytes, these lamp brush chromosomes we are observed of the salamander. So in which stage meiosis means? In which stage? Whether the prophase, then prophase consists against 5 subphases, you know. Which type of subphases? Which type of? Leptotin, zygotin, parkitin, diplotin, diakinesis, you know. Are you forget?
Yes, sir.
These are the 5 subphases we are observed in the prophase of the meiosis. So meiosis 1 prophase consists 5 subphases. They are leptotin, zygotin, parkitin, diplotin, and diakinesis. So these lamp brush chromosomes we are observed or we are found in the amphibian oocytes at this time of prophase. In the prophase, diplotin subphase of the meiosis, we are found these lamp brush chromosomes. Is it clear?
Yes, sir.
Is it clear? And my dear friends, here in this Lamb-Brush chromosomes also in haploid conditions. Why? Because it is these chromosomes absorbed in oocytes or reproductive cells. Always after meiosis, definitely the diploid state is going to form haploid conditions. So these Lamb-Brush is also haploid conditions, whereas these salamanders of haploid chromosomes, these loops, these how many loops are we are observed? DNA loops. So more than 10,000 loops we are observed in the Lamb-Brush chromosomes of the salamander. So this image shows about the Lamb-Brush chromosomes of amphibian oocyte. This is an immature egg. So this immature egg shows this Lamb-Brush chromosome. So I will draw this Lamb-Brush chromosome. See here. So these are the, so this is loop. So this type of number of loops we are observed, on the chromosomes. These loops looks like a brush-like structures. So that's why these chromosomes also we can call as Lamb-Brush chromosome. So this image we are observed in the ultra structure. So we are observed in the electron microscope. This picture about Lamb-Brush chromosome. So these are the loops, So this is the chiasma. Chiasma means X-shaped structure in the division of the surface, like pocketing. This is the third surface of the prophase 1 or meiosis 1 of the prophase 1. The third surface, that is the pocketing stage, we are observed chiasma. So number of chiasmas we are observed in this loop. See here, this is the one chiasma and this is the one chiasma, and this chromosome shows having number of loops, So loops where active RNA transcription is occurring. So loop region is the very active region for transcription regions, or transcription has occurred in the regions of the loops. So these loop region, so this is the loop, This is the loop. So this loop is the region which is a very active region for transcription. What is the meaning of transcription? Anybody can tell? Transcription means transcription. Have you ever heard this term transcription? DNA present in nucleus, DNA present in nucleus. From DNA to mRNA, the synthesis of mRNA, this the important stage we can call as transcription. So this process depends upon the transcriptase enzyme. So transcriptase enzyme is useful for the process of transcription or mRNA formation or synthesis of mRNA. So this is also we can call as DNA dependent RNA. So mRNA we can also call as DNA dependent RNA. So this transcription we are observed in the nucleus region. From mRNA to protein. So mRNA to protein synthesis we can call as translation. Am I clear? Translation. So this translation we are observed in the cytoplasm of the cells, whereas the prokaryotic living organism or eukaryotic living organism, whether the unicellular organism or multicellular organism, so translation should be in cytoplasm. But remember, my dear friends, whereas the prokaryotic living organism, transcription and translation both takes place in cytoplasm only. Am I clear? I hope this topic is completed.
Yes.
Transcription and translation.
Yes, sir.
So this transcription has occurred in the part of the nucleus, whereas translation we are observed in cytoplasm. So transcription means from DNA to synthesis of mRNA. So this mRNA depends on DNA, or DNA dependent RNA we can call as mRNA. So synthesis of mRNA we can call as transcription. From mRNA to formation of the protein or synthesis of the protein we can call as translation. So this translation process entirely process are entirely present in cytoplasm. But if the prokaryotic living organism, transcription and translation both are present in cytoplasm only. Is it clear? But some of the viruses, like this pandemic situation caused by which virus?
Coronavirus.
Coronavirus. Corona is a disease virus, you know. SARS-CoV-2. So these DNA to mRNA we can call as transcription, you know. Transcription. Is it clear? But whereas DNA is the genetic material of living organism, but our SARS-CoV-2 or COVID virus, the genetic material is, anybody can tell?
RNA.
Very good, my dear friend.
RNA.
Single stranded RNA. Okay, single stranded RNA. So this RNA is act as a genetic material in virus, that is the COVID virus. Along with COVID, HIV virus is also RNA virus, you know. Polio virus is also RNA virus, you know. So this RNA virus, so already RNA is there. Then how this transcription takes place? So that's why RNA, from RNA to DNA is produced.
It takes up the host DNA.
Amma? Once again. Amma, tell me, don't worry. Correct?
It
So
takes
this
up the host DNA, sir.
Yeah. So this RNA, not mRNA, from RNA to DNA synthesized process we can call as reverse transcription. Am I right? Yeah. The enzyme is reverse transcriptase. So that's why RNA viruses does not control by the usage of the normal drugs. So normal drugs unable to control the viral diseases like HIV. Am I right? Like corona. So that's why these the reverse transcription process or reverse transcriptase is an enzyme present in the viruses. So that's why they are not controlled by the normal drugs. So reverse transcription, the reverse transcriptase. So this is the process we are observing the RNA viruses, not DNA viruses, my dear friends. So DNA, generally viruses are contained only one type of like RNA or DNA, whether they are possess RNA or DNA. So whether the DNA consists that viruses we can call as DNA viruses. If they are possess RNA genetic material, so that viruses we can call as RNA viruses. Okay. I hope you clearly understood.
Yes, sir.
Okay, very good. So then these lamp-breast chromosomes also we are observed not only in amphibians but also acetabularia. The largest chromosome. My dear friends, in competitive exams we are observed the largest or longest chromosomes we are observed in the acetabularia plants. So this acetabularia plant possesses the largest or longest chromosome. Highly largest chromosomes we are observed in the plants. The plants are acetabularia. Am I clear? Acetabularia. So this acetabularia chromosome is also lamp brush chromosome. Very, very important, my dear friends. Lamp brush chromosomes. Clear? Then the last one. And last one, the third type of chromosomes, we are called B chromosomes. Compared to Polytechnic chromosome, compared to lamp brush chromosomes, B chromosomes are very, very smaller. Compared to normal chromosomes, B chromosomes are very, very smaller. So that's why B chromosome not belongs to GAINT chromosome or GAINT chromosome. GAINT chromosomes only two. Number one, Polytechnic chromosome, also we can call as salivary gland chromosome. First discovered by Balbioni in 1881 in Diptera, or the insects salivary glands like Drosophila melanogaster salivary glands. So that is the first type of GAINT chromosome or GAINT chromosomes. And the second type of GAINT chromosome or GAINT chromosome is lamp brush chromosome. These lamp brush chromosomes were observed in oocytes of the amphibians, for example, salamander. And the third type of chromosomes, the chromosomes are not related to normal chromosomes, not related to GAINT chromosomes, but the size of the chromosomes is very smaller compared to normal chromosomes and GAINT chromosomes. Generally, these B chromosomes are identified only plant species, not animal, not human beings, only flora or plant species. The best examples of the B chromosomes we are observed in geomes or corn plants. Okay, corn plants, geomes or corn plants. So this corn plant or geomes belongs to which family? Okay, Graminae family. In earlier, very good. In earlier we can call it Graminae, but according to ICBN, the family name should be AC. So this Graminae we can now call as Poaceae. So the geomes contain 200. How many? 200 species. Not only along with these geomes, but also some of the other organisms, lower plants like Bryophyta and Pteridophyta, which is also possess these B chromosomes along with gymnosperms also. So Bryophyta, Pteridophyta, and gymnosperms, both they are possess these smaller size chromosomes or B chromosomes. These B chromosomes are not clearly explained, not clearly observed. Still now, the research is going on about B chromosomes. Okay, and my dear friends, the B chromosomes is very smaller to other chromosomes, and these are identified only plant species only, not animal species. But whereas the Polytechnic chromosomes and the lamp brush chromosomes, both they are identified all the living organism along with plant species and animals also. Okay, so this is about the special type of chromosomes. Special type of chromosomes are three. Number one, Polytechnic chromosome. Number two, lamp brush chromosome. Number three, B chromosome. First two chromosomes are very compared to larger size chromosomes compared to normal chromosomes. That's why the chromosomes we can call as GAINT chromosomes or GAINT chromosomes. And the third one type of chromosomes are compared to normal chromosomes, they are smaller size or length of the chromosome compared to normal. The size of the B chromosome is smaller size. So B chromosomes only identified in plant species, not any animals or human beings, but only lower organism or lower plants like Bryophyta or Pteridophyta or gymnosperms. Okay, along with the grasses like Graminae family or Poaceae family, the best example geomes, corn plants. Okay, so this is about the special type of chromosomes. If you have any doubt, please ask me. And this is the time for your question or queries. Okay. Any doubts? Any doubts, my dear friends?
No, sir, it's clear.
It's clear. So you are very good students, because you are the one of the prestigious institution, belongs to prestigious institution. That is the Reddy College, Okay. So that's why you are very well known about these special type of chromosomes and the structure of the chromosome, chromosome number, and shape of the chromosomes, types of the chromosomes. Okay. So yesterday and today we are clearly explain about studied about the chromosome shape, size, structure. Okay, structure and types of the chromosomes and number, chromosome number also, whether they are euploids or aneuploids. Whether the euploids, they are haploid condition or diploid condition or triploid condition or tetraploids. Whether they are aneuploids, monosomic, nullisomic, trisomic, and tetrasomic condition. Okay, the types of the chromosome based on the anaphase. Based on the anaphase, we are observed 4 types of the chromosomes. That is metacentric, submetacentric, acrocentric, and telocentric chromosome. Okay, so these are the 4 types of the chromosomes we are observed, whether the mitotic division or meiotic division, anaphase. Especially anaphase, we are observed this type of the chromosomes based on the position of the centromere. Chromosomes are metacentric chromosome, submetacentric chromosome, acrocentric chromosome, and telocentric chromosomes. And the special type of along with these normal chromosomes, special type of chromosomes also we are observed in this, different living organisms such as Diptera insecta, Diptera, along with amphibians, and some of the other plant species. So compared to normal chromosomes, larger size chromosomes we are observed. The larger size chromosomes we can call as GAINT chromosomes or devil chromosome. Okay, so these GAINT chromosomes are 2. Number 1, Polytechnic Chromosome, also we can call as salivary gland chromosome, identified by or discovered by Balbioni in 1881. The second type of chromosome, GAINT chromosome is lamp brush chromosome, identified in 1981 in the amphibians, oocytes of the amphibian. The best example of this oocytes of the amphibian is salamander. Okay, so this is about the GAINT chromosome. Then what about the smaller chromosome compared to normal chromosomes? That is B chromosome. So B chromosome is very smaller size chromosomes, which is only identified in plant species only, like the lower plants like Bryophyta, Pteridophyta, and Gymnosperms. Along with these lower plants, some of the monocots like Graminae plants or Poaceae family plants like Geomage possess this B type of chromosomes. So these B type of chromosomes also special type of chromosome. Okay, this is about the special type of chromosomes, and not yet clearly explain about these B chromosomes, but research is going on still on B chromosomes. So from Reddy College, like you, my people or my friends, is going to clearly research on that B chromosomes, and you will get the Nobel Prize. My wish. Okay, my dear friends. So this is today's class. Tomorrow we will go into sharp 8 p.m. By another topic, that is the cell cycle or cell division. Okay, mitotic and meiotic cell division is there. Better I will stop here. If you have any doubt, please ask me, my dear friends.
Girls, if you have any doubts, please be free to ask to sir. Sir is very, very free to us. Without hesitation, sir can help out in all our doubts. Okay? Feel free to ask sir if any doubts are there.
Okay, very good students. Because of Kavita Madam students and Dr. Rajni Madam students are very clever and very intelligent students. They know very well and they are listen very clearly and they are understood very clearly. So that's why they are not able to speak even single word. But they know very well. They are very clearly understood. I hope.
It's not like that, sir. Once if they start reading, then they'll come out with very lots of dots. For now, they are just
Then
listening.
who starts? Who starts questioning? Any queries, my dear? Pujita, Pravalika, Reddy, Srivani.
Yes, ma'am.
Okay, very good.
Ananya,
Good response.
Are you there, Ananya?
Arpita, Ananya, Sai, Pujita.
Ananya, Renu, are you having any doubts, ma?
No,
Maheshwari.
ma'am, it's clear, ma'am.
Yeah.
Yeah.
Thank you.
And what about in FNBC?
Yes,
Selva.
ma'am.
Only 51 participants are there. Among these 51 participants, no one have any doubt.
So you can post questions.
Sir, you can ask questions.
Okay. Okay, anyway, thank you, my dear friends. Tomorrow, sharp 8 p.m., we will meet in our Google Meet through online. Thank you.
Sir,
Thank you, my dear friends.
Yeah,
Thank
yeah.
you, sir.
Thank you very
Thank
much,
you,
sir.
sir.
Thank you, Dr. Kavita Madam Garu. Thank you, Dr. Rajni Madam Garu. And
Thank you,
we
sir.
will meet by tomorrow. Okay? Evening. Thank you.